You might have seen headlines like this about Silicon Valley trying to create perfect baby. That's because people like Sam Altman and his husband, and crypto billionaire Brian Armstrong, and many others have been funding a new group of startups. And they're all working towards one goal: editing the DNA in human embryos, which really opens up a can of ethical worms. But to me, all of this money and effort misses what's most exciting about gene editing. So I talked to researchers from 4 different UC labs. They're using CRISPR in really creative ways to tackle some of our world's biggest problems. And one of these labs showed me how CRISPR is starting a new medical revolution. Gene editing isn't new. Tools like TALENs and zinc finger nucleus have existed for decades, but they've been kind of clunky and unreliable. But in 2012, a new, more precise gene editor came on the scene. Jennifer Doudna and Emmanuel Charpentier are CRISPR's co-inventors. And the crazy thing about their work is that they weren't trying to create a new gene editing tool. But during one of their experiments, they had an epiphany that would change the field of genetics. And it got scientists very excited.
2018, I walk into Jennifer Doudna's office in this building, the Innovative Genomics Institute. And I sit down before Jennifer and say, "Jennifer, what a treat to see you. What are you trying to do with this Innovative Genomics Institute thing?"
This is Fyodor Urnov. He now works for Jennifer at her UC Berkeley Institute, which is coming up with the life-changing uses for CRISPR that we'll learn about in this video. Fun fact, he's also the guy who coined the term gene editing. Although his gene editor was like the Model T, CRISPR is like the Enterprise. And here's the thing that has Fyodor so excited about CRISPR.
Jennifer, with a calmness and a seriousness that I will never forget, says to me, "Fyodor, I want to make CRISPR gene editing the standard of medical care." I tried to hide my surprise. I don't think I did. It's an extraordinarily ambitious statement. And here we are, 2025. We've made formidable strides.
We're going to unpack all of that in a minute, but before we do, we first need to understand how CRISPR gene editing works.
It's called gene editing for a reason. If you think about a word processor, you click on a typo and you hit backspace and you type the correct text. Now it reads well. That's what CRISPR gives us for genetic diseases.
Sounds simple enough, but the technology behind creating this life-changing tool is as complicated as it is fascinating, because it's been hiding in plain sight for millions of years. And it all starts with a microscopic war that's been raging between viruses and bacteria. Viruses and bacteria are constantly fighting. Viruses inject their DNA into bacteria, which makes more viruses, which means viruses outnumber bacteria by a lot. So why haven't they totally won out? Bacteria evolved a way to fight back. See, back in the 80s, Japanese scientists started sequencing bacterial DNA. And they saw something no one had ever seen before: palindromic repeats in DNA, like the word race car that reads the same forward as it does backward. This was strange. Why were these repeats there? It wasn't until 20 years later that a lab in Spain would find the answer. See, when bacteria and viruses fight, bacteria saves a chunk of viral DNA between the repeats. When the virus invades again, the bacteria can now recognize the intruder and deploy a special protein like Cas9 to cut up the viral DNA. Now it made sense. This was a bacterial immune system, and a Spanish lab named it clustered regularly interspaced short palindromic repeats. But they shortened that to CRISPR. But how do we go from bacteria fending off viruses to humans using it to edit DNA? That's where Doudna and Charpentier's labs come in about 10 years later. See, DNA makes a molecule called RNA that basically tells our cells what to do. And each RNA sequence is a unique but exact match to the gene that produces it. The bacteria's immune system uses RNA to track down the invading virus and attach to it, then chop it up. So Doudna and Charpentier figured this: scientists could create a guide RNA sequence to find any gene, then attach it to the Cas9 protein. This combo can find its DNA target and make a cut. After that, Mother Nature can repair the cut itself, or scientists can tell DNA how to repair it, eliminating the genetic typo. Or
click on a typo and you hit backspace and you type the correct text. Now it reads well.
Doudna and Charpentier basically took this tool from nature, improved it, and turned it into a tool we can use to edit the DNA of any life form. And in 2020, they won the Nobel Prize in Chemistry. This award was betting big on CRISPR's potential to change and even save people's lives. And what I love about this story is that CRISPR began with scientists asking a simple question. Why do bacteria have this weird repeating pattern in their DNA? Critics often complain about science that doesn't have a clear goal or a business plan.
What it says to me is they have too much money if they're going to spend money on something like that.
But that's exactly where real discoveries come from. Doudna and Charpentier didn't set out to invent anything. They were following a mystery, and in doing so, made a world-changing breakthrough. Once CRISPR became available to scientists, it gave them a powerful new tool to solve some of our biggest problems. And that brings us to the first lab I visited, which is trying to save the chocolate industry. That's right, the world's chocolate supply is under threat. In severely affected regions, up to 50% of cacao harvests are being wiped out by a single pest called the cacao swollen shoot virus. These sweet-tooth mealybugs spread the virus from tree to tree. Pesticides aren't that effective against the mealybug, and there is an experimental vaccine, but it's expensive and produces smaller harvests. So right now, the main way to fight the virus is to simply cut down the infected trees. So to save the $100 billion chocolate industry, Mars Inc. is betting big on CRISPR.
So let me introduce you to my colleague, MJ. He's the Michael Jordan of tissue culture.
Myeong Je-Cho and Brian Staskawicz at UC Berkeley are experts in plant biology, and their lab is using CRISPR to do exactly what it evolved to do: find a virus and kill it. But to do that, the research team had to do something incredible. You can't just inject CRISPR into a tree and let it find the virus. Like humans, plants have immune systems. Our white blood cells move around our bodies, but plants' immune systems are stationary inside every cell. So the cells that regenerate the tree all have to be equipped with CRISPR.
We can see embryos here. Basically, these are derived from single cells.
That's right. They had to start with a single cell, then figure out the perfect blend of nutrients and conditions to grow it and grow it and grow it into a fruit-bearing tree, which takes 5 years. I have enough trouble keeping a houseplant alive, so the fact that they're growing trees from a single cell blew me away. The next step is to ship the trees off to Mars Inc. in Davis, where they'll be tested against the virus and see whether this all works. Rolling out something like this comes with all kinds of challenges. For one, the world's entire cacao population would need to be replanted with these trees. The good news is you can splice off a piece of CRISPR tree and grow more from it. This isn't the only way CRISPR could change farming, though. There's the potential for bigger harvests and more nutritious crops. And right now, UC Berkeley's Innovative Genomics Institute is using gene editing to make rice plants more drought resistant. But our second lab looks at a new frontier for gene editing, one that was nearly impossible to predict when CRISPR was invented. And it's helping to develop a brand new treatment for the most common chronic disease in children.
We now have the ability to use CRISPR to edit entire populations of tiny microbes called microbiomes that live in and on our bodies.
Scientists are just beginning to understand how our microbiomes affect our health, and they now believe they're one of the main drivers of something we never expected: asthma. So researchers are trying to stop the damage it causes to our airways. See, when viruses attack, they destroy cells in our noses. This creates a feast for bacteria, which rush in and cause inflammation. So in an NIH-funded study, Sue Lynch's lab at UCSF looked at asthmatic kids' microbiomes while they had an upper respiratory infection, and they discovered a telltale pattern. Kids who didn't experience an asthma attack had 3 kinds of microbiomes in their noses, and in kids who did have an attack, 3 totally different microbiomes. But why do these microbiomes lead to asthma attacks while the others don't?
It turns out that the pathogenic bacteria in the airways of these children turn on genes that make a molecule that constricts the airways, and that is what leads to asthma exacerbation.
So turn off those genes, stop asthma exacerbations. This is where CRISPR enters. Or actually, that's the problem. One thing I kept hearing from scientists: CRISPR is incredible, but it has one challenge: delivery, delivery, delivery. So how is Lynch's team getting CRISPR into these bacterial cells?
We're considering a kind of Trojan horse approach.
Bacteria share DNA. They do it, for instance, to boost their body's antibiotic resistance. So Lynch's lab wants to arm good bacteria with CRISPR, then introduce them to the bacteria that promote asthma attacks. The two will swap DNA. Once CRISPR is in, it'll turn off the gene that causes asthma exacerbations. Now, the bad bacteria can be replaced by the good bacteria. This research is incredible to me because we're turning bacteria's own immune system against itself. And the researchers are looking at the microbes in infants' guts to find ways to prevent asthma from ever even developing. Beyond human health though, microbiomes can cause all sorts of problems. And our third lab shows how cow burps are a big part of that. Scientists studying cow burps sounds like one of those wasteful research projects, but it's so far from it. Cow burps contain mainly methane, which is a really potent greenhouse gas. Multiply all of that methane by all of our cows, and that makes about 5% of the world's annual greenhouse gas emissions. So Ermias Kebreab and Matthias Hess's labs at UC Davis are working to find and eventually edit the microbes in cows' guts that make methane. To do that, they're putting cows on an unusual diet. Years ago, this farmer made a surprising discovery. By feeding seaweed to his cows, they burped a lot less. Since then, Kebreab's lab has shown the seaweed diet can reduce methane emissions by up to 80%. So why not just feed every cow seaweed? Most cows graze in fields, so it's hard to get the seaweed to them. Plus, they'd have to be on this diet all the time. But with CRISPR, they want to change a cow's microbiome for good.
That's what we're trying to figure out here, and then also obviously to isolate some of the genes that are responsible for methanogenesis as well.
That's not easy. Microbiomes are complex ecosystems composed of thousands of different interdependent microbes. Plus, they're hidden inside the cow's gut. So to find the right microbes, researchers have an unusual tool.
Here is replication of a cow gut. In the machine behind me, we have actually 12 of these vessels.
Just add a bit of stomach fluid from a cow and different mixes of seaweed to simulate how the microbiome changes when methane is reduced. Once researchers figure out which microbes are responsible for all the burps, they'll use CRISPR to disable the methane-producing genes. Then they'll introduce that new microbiome colony into the artificial cow gut to test it out, before trying it in a real cow. Now we need to do this treatment for all the cows on Earth today, but the hope is that mothers would pass along their new microbiome to their calves. And doing that would have a huge impact on our fight against global warming. These three projects are all ambitious, and we'll have to wait and see how they turn out. But CRISPR is already changing people's lives, because there are people walking around today who have had a debilitating disease treated by CRISPR. Which brings us back to Fyodor and the goal of turning CRISPR gene editing into the standard of medical care. That's happening thanks to this last lab. Human DNA stretches around 3 billion nucleotides long. But if just one nucleotide pair is changed, you can get sickle cell disease. Some red blood cells develop a sickle shape and can cause a ton of terrible side effects. But so far, CRISPR has stopped some of the worst symptoms and the progression of the disease for over 200 people by changing the mutated gene that produces hemoglobin inside our red blood cells. And earlier last year, scientists diagnosed a newborn with a rare genetic disorder that has a 50% fatality rate. But Fyodor and a team of collaborators from around the world custom built and delivered a CRISPR treatment for the baby. In case you're wondering, there's a big difference between this and editing embryos, like what the Silicon Valley companies are trying to do. This kind of work has restrictions or bans in the US and many other countries. In fact, in 2019, a researcher in China was sentenced to prison for secretly creating the first genetically edited babies from embryos. He tweaked their DNA to give them HIV resistance. And that might sound like a great idea, but scientists are largely against this work because it's so risky. We're still learning so much about our genes, and if a mistake is made, we may not know until years later. Plus, any errors would be passed down to future generations of children. While many of these companies say they're working on curing genetic diseases, some scientists worry that we're really heading towards creating designer babies, where wealthy parents could tinker with their kids' looks, or intelligence, or athletic abilities. The treatments that Fyodor works on are different. Once someone has been born, any edits made to their DNA aren't passed down to future generations.
300 million people have one of 6,000 genetic diseases, and pretty much the vast majority are genetic typos. That's what CRISPR is purpose-built for.
This is the start of the standard of medical care that Jennifer Doudna envisions. custom designing CRISPR for anyone with a genetic disease.
So the vision is this. First, build a process where you can make a CRISPR on demand, treat a child before the child dies or has severe disability. Step 2, say to the clinicians, do you have children with a related disorder? Also immune system, but not exactly the same one. Well, do we have a CRISPR on-demand process for you?
That sounds like a really big endeavor. So how would that actually work? Fyodor has a great analogy.
You go to a pizza place and you ask for something with a different topping than the previous customer. Swapping out the topping is trivial. Why? Because there is a platform for pizza baking. What does that mean in English? Well, it's the cooks, the person who throws the disc, the oven, the sauce, the cheese. You just swap out individual ingredients. Can make pizzas on demand. The same with CRISPR. Literally, the analogy holds. A platform for CRISPR cures would have stereotypical enabling components, the scientists, the physicians, the hardware, the manufacturers, the interface to the regulators. What would be made on demand, if you will, is the topping, namely the bit of CRISPR that routes it to the new gene. We're eager to enable this in the US healthcare system. Our regulator, the FDA, is welcoming this concept, where we can build a platform for genetic cures on demand.
CRISPR's future looks promising. It's being used in clinical trials to treat everything from high cholesterol to UTIs to neurodegenerative diseases and currently untreatable cancers. But here's CRISPR's big secret. We never would have created it without federal funding for basic science, the kind of research with no immediate payoff. See, after World War II, America invested heavily in this type of research.
No country has ever been so bold as to say, these scientists, these people in white lab coats, let's give them some money. Let's see what they come up with. No other nation in history has made such an investment in fundamental innovation that creates staggering biotechnology breakthroughs that improve human health.
In fact, 99% of FDA-approved medication comes from federally funded research. But over the years, this federal investment has been declining. Science takes time, and it's not always clear which research will lead to the next big breakthrough, which makes it hard for private companies to invest in basic research, like what led to CRISPR. Take a look at this jellyfish. In the 1960s, a marine biologist was given federal funding to understand what makes it glow. This might seem like a pointless question when we could instead fund nothing but cancer cures or heart disease treatments. But the answer to this simple question about a jellyfish completely revolutionized medical research. Check out our video all about this and why pointless questions are so important. And if you like stories about science and discovery, be sure to subscribe to Fig1.